segmented worms, and you're going to see the characteristics of phylum Annelida, the different classes, and then we are going to have one type study which is about the Indian cattle leech or Hirudinaria. Its hemocoelomic system, the adaptations shown for the parasitic life, and what exactly is metamerism, what is schizocele, what is a closed circulatory system, the importance of it and evolution in this particular area per se. And then you'll also be seeing the different kinds of excretory organs in different annelids. Let me share my screen with you. So when it comes to phylum Annelida, you get to see that, it includes the earthworms, the leeches, different kinds of blister worms, etc. So in this, again, vermis means it means a soft-bodied animal, basically. And then it also has parasitic worms. Some of them, a few are arachnids. Arachnids belong to the spider family, and even crustaceans. So Lamarck, it's not landmark, it is Lamarck actually. He had given the term annelida so that it means annelida means like it is ring-like. Annulus means ring, and form means idus means form. So in this, what is happening is these rings help, and this is the metamerism that is being exhibited. So nephridia that are present, those are the excretory organs. They're ectodermal in origin, and they are different in sizes, positions, the nature, and the kind of from where they are opening. You also have Celomo ducts. They are again mesodermal in origin. You're going to learn about it in some time. And in these, some of the animals fuse with nephridia. So what happens is there is discharge of both excretion, and at the same time, there is even gametes that is coming out of the pores, basically. I request you all to mute yourselves. You'll be able to hear me properly and clearly. Right. So when it comes to the general characterizations, you get to see that these are all like, they have a fluid-filled coelom, wherein which acts as a hydrostatic skeleton. This is useful for burrowing and locomotion. You have, they are like, cephalization is more pronounced. That is, you have a distinct head, which has tentacles. You have some eyes on them. And there is metamerism, that is, the body is divided into a number of ring-like segments or metameres or somites. These segments are separated from each other externally by transverse grooves, internally by septa. Septa is some small partition of the body wall that comes in, so as there is a distinction, and there is a like you can see that septa is something like your room wall, basically. So there is a partition inside the body as well. And this kind of segmentation is called as metamerism. In some animals, this external segmentation corresponds to or is it is in lieu with the internal segmentation. Example is Ferritema. Ferritema is your earthworm. And in some like leeches, the segmentation is only present externally. So annelids are in different forms. They can be like, marine, freshwater, terrestrial forms. Most of them are free living. Some of them are burrowing. Some of them live in an ectoparasitic life. And so the body is divided into three regions: prostomium, trunk, and pygidium. Prostomium is the first, the pre-oral segment. Then the first segment is known as the peristomium again, and the anal segment is called as the pygidium. In between, you have the trunk. Body wall is thin, transparent, non-cellular, called as the cuticle, composed of different kinds of muscles. And you have two different kinds of muscles that are present, which is, the longitudinal muscles and the circular muscles. And then you have chitinous bristles that come out of from the epidermis. They are known as the setae, and they help in, giving a grip, or, they'll be able to hold the substratum with the chetae or the setae. And again, these are the animals that use setae as the locomotory organelles. And all the annelids are coelomate. wherein the body cavity is a true perivisceral, I mean inside the visceral coelom, which lies between the two mesodermal layers. So coelom is again filled with coelomic fluid. This acts as a hydraulic skeleton, and in leeches, the coelom is very much reduced. Alimentary canal is present right from mouth to digestive system. So you have a complete digestive system present, and it has, it is a muscular type in Annelida. Digestive glands are also present. Diffusion, for diffusion, is the process through which the exchange of respiratory gases, oxygen, carbon dioxide takes place. That is through the body walls and the gills. That is one of the reasons for which they require moist surfaces or moist places to live in. In many polychaetes, parts of the parapodia are modified into gills, especially in the marine or in the ones that are present in aquatic environment. And annelids are the first animals with a well-developed closed blood vascular system. And hemoglobin is the respiratory pigment. So you need a respiratory pigment to carry the oxygen or the respiratory gases. So annelids, from annelids onwards, you have different kinds of respiratory pigments that is present. So one of them is what do you call it? Hemoglobin, which is present. So this is present dissolved in the plasma of the blood. And excretory system is again made up of metanephridia, nephridia, and they are metamerically arranged, coiled ectodermal tubes. So they communicate with the coelom and the exterior for the necessary excretory function. And you have a nervous system which is consisting of a circumpharyngeal ring, that is around the pharynx, and a ring and a ganglionated double ventral nerve cord, that is the nerve cord which is present on the ventral side. And this since it is present around the pharynx along with the supraesophageal ganglia, this is actually known as a nerve ring. And then ganglia extend throughout the length of the body. And sense organs are tactile organs, taste buds, eyes, such are present, and they are like photoreceptors, chemoreceptors. Photo, as you know, light receptors. Chemo for for sensing, and mechanoreceptors. So the same thing is given in your textbook again. And most of the annelids are bisexual, and that is they are monoecious in form. And then, the bisexual ones forms have the gonoducts also. In polychaetes, sexes are separate and gonads develop from the coelomic epithelium. Gametes pass out through the nephridia or through the Celomo Ducts. Development is direct in the monoecious forms, that is the oligochaetes and the hirudinaria, and indirect in the dioecious forms, such as the polychaetes and the archaeannelids. So in the indirect development, obviously, since the term itself, you can easily guess, you have a larval stage, and the larval stage, the larval form is the trochophore or the trochosphere larval form. And cleavage, whenever the zygote is present, how is it going to be having a cleavage? So it is a spiral and a determinate. That means it is going in a determined manner. So this is what you mean by determinate. And when you see reproduction, it is asexual by budding, especially in some. Example is Psyllis ramosa as given. So when it comes to the classification of phylum Annelida, you have around 8,700 species, and so that's what this number depends according to the different places that you get to read about them. So again, the phylum Annelida is divided into 4 different classes, which is polychaetes, oligochaeta, hirudinia, and archaeannelids. So different examples are again given accordingly, and these are the 4 main classes based on the presence of or absence of parapodia, setae, metamias, and other morphological characters. So also attempts by 2 different scientists and others have been seen over here to group in the class heterogeneous and for so you have basically 2 different classes in the class polychaeta, that is Erantia and Sedentaria. So these are the two different subclasses as seen in class Polychaeta. So poly means many, chaetae means hair. Also commonly called as bristle worms, and they are the most diverse group, having different kinds of creatures in this particular group itself. They're mostly marine, some of them are freshwater forms, and segmentation is present both internal and external, with a distinct head, eyes, and palps and tentacles present. Again, poly, so it means numerous setae are present on the lateral parapodia, that is the parapodia are meant for locomotion, and they exist as bundles. So again, as you see, some of them are ectoparasites also, or that is they live on other marine crustaceans and fishes, and almost all of them are carnivorous in nature. Body cylindrical, elongated, segmented, different kinds of similar segments are present. You have eyes, tentacles, cirri, palps, mouth, and the last segment is again called as the pygidium. And you have lateral outgrowths of the body which are known as the parapodia, and these parapodia again have different kinds of numerous setae present arranged as bundles, because of the name because of which it got the name Polychaeta. Now each parapodium is again divided into a natopodium and a neuropodium. You can probably see that in your textbook figure 7.1. So if you see the figure B, you can get to see that, the fourth point, you have different kinds of the third and the fourth in the diagram. So each is again divided into, each parapodium is divided into a natopodium and a neuropodium. And these are the organs for locomotion as well as respiration. Now a clitellum is absent and the sexes are separate, and most of the segments have gonads, and this is present in many species of the classes. And if not, the gonads are absent or temporary as seen in some species. Now the gametes are shed into the coelom and then they are spawned through the metanephridia. And if you see here, the body wall again is dermomuscular with thin, tough chitinous, non-cellular cuticle surrounding it. And again, you have musculature, which is what do you call it, longitudinal and circular muscles, both of them are present. Respiration is by skin. Alimentary canal is straight. So you have from mouth to anus complete alimentary canal present. And some of them are mucosal feeders, some of them are filter feeders. And you get to see that there is some, in the case of some polychaetes, example is Aphrodite, the setae are modified to extract dissolved oxygen from the water. And such kind of setae are known as elytra. And in some polychaetes, these setae are modified as gill-like structures. So that is, example is Arenicola. Now again, blood vascular system in the whole of annelids is a closed type. So you have definite blood vessels and you have hemoglobin dissolved in the plasma. Excretory system has paired nephridia arranged metamerically. Nervous system is again basic thing with the ventral, pharyngeal uh, nerve ring, and again dioecious, that is the sexes are separate. And as I already told you, sexual and non-sexual parts of the body are completely distinct. And the non-sexual part, that is, the non-reproductive part of the body is called as atoke, and the sexual or the reproductive part of the body is known as epitoke. Now during the breeding season, what happens is the epitoke gets separated from the atoke. Then this starts leading an independent existence till the gametes are released outside. So for example, in some species like Silus and Hyena, the epitoke develops a head and becomes an independent male or a female. So gonoducts are absent. Fertilization is external. Cleavage is spiral determinate. Development is into trochophore larva, which undergoes metamorphosis to form the adult. And asexual reproduction is also seen by serial or lateral budding, as in the and the examples of this class Polychaeta include Chaetopteris, Arenicola, Neanthus, which is Nereis, Silis, and Aphrodite. Now, when we come to the second class, that is class Oligochaeta, we get to see that these are terrestrial earthworms. Quite a number of freshwater and marine forms are also present. Segmentation is corresponding to it is present both external and internal, and also you get to see that these they are corresponding to each other. What is present outside is present inside also, and appendages are absent in these animals belonging to this class. So again, head is not exactly so distinct without the appendages. So you get to see that the prostomium is very small without any kind of eyes or tentacles or parapodia, and chitinous setae are also present according to the different kind of segments that they are in. And then they usually S-shaped with a middle swollen nodulus. And in earthworms, the party the body is thickened without segmentation, and this is known as the clitellum. And body wall is dermomuscular. Same thing again, you have both circular and longitudinal muscles. And body cavity is the true coelom, which is lined by the coelomic epithelium, which is mesodermal in origin. And then you have transverse septae, which divides the coelom into the compartments. And again, coelomic fluid is present. This causes or acts as the hydraulic skeleton for the complete organism. And then you have the coelomic fluid, which comes out through the dorsal pores so as to keep the skin moist for respiration, because respiration is through the gill slits and the skin. And longitudinal locomotion is by contraction and relaxation of the longitudinal muscles and the circular muscles. And setae also play an important part in the locomotion. And alimentary canal again is from mouth to anus, gizzard oval, which is a part called as a gizzard is present, which is a thickened part of the alimentary canal. It is used as a grinding organ. Respiration occurs through the body surface. Blood vascular system is closed. Dorsal, ventral, subneural blood vessels, lateral hearts, all of these are present. Again, hemoglobin is the respiratory pigment which is present dissolved in the plasma. Excretory organs are the nephridia. And aquatic forms excrete ammonia. Terrestrial forms excrete urea. So aquatic forms are ammonotelic. Terrestrial forms are ureotelic. And a special feature is that you have got some cells which are known as chlorogogen cells. These are present around the intestine, and these cells play a major role in the excretion and also in the intermediary metabolism. So you can compare these chlorogogen cells to the liver of the vertebrates. And again, the nerve ring is present. You have the circumesophageal connectives also present. Ganglionated double ventral nerve cord. Sense organs are poorly developed. They are, and when you come to the oligochaetes, they are hermaphrodites, and the gonads are distinct with gonoducts, and they're only present in the genital segments. Sorry?
Polydeveloped.
I didn't get you. Yes?
Polydeveloped.
Where is it there? Can you please tell me?
Polydeveloped, ma'am.
Can you please type it? I'm not able to understand what you're telling.
Sense organs.
Sense organs are poorly developed. That means that, it is like, the sense organs are not very highly developed. We have got extremely good sense organs. And in case, when you compare it along with the polychaetes have different kinds of sense organs that are present as compared to the oligochaetes. And then you can get to see that, you do not have those tactile receptors. You do not have proper eyes, tentacles, and you do not have what do you call it, very good did you get it? The taste buds, eyes, tactile receptors, and all these are not exactly very well developed in the oligochaetes. Okay,
thank you, ma'am.
Yeah, right. So where was I? Right, development. So what happens is, oligochaetes are hermaphrodites. So and that is their monoecious. And then the gonads are distinct. You have got gonoducts also. Reproductive cells mature in the seminal vesicles or in the ovary sac. And the gonads are present only in the genital segments, as you see, as I guess you have all studied earthworm in your intermediate. So you get to see that they're present only in the clitellar segment. And then the development is direct without a larval form. Fertilization is when, there is a cocoon that is discharged, and the ova and the spermatozoa are discharged into that particular cocoon that is secreted by the clitellum. And then in that clitellar segments only fertilization occurs. So it is an external type of fertilization. Now what happens is direct development is direct. So there is no larval stage. Young ones come out of the cocoon. So in the aquatic oligochaetes, what happens is asexual reproduction is also present. And then they have a high power of regeneration. You have already seen that. So example is Megascolex, Ferritima. All these are earthworm species, Lumbricus and Tubifex. So let's come to the third class, which is the Hirudinea. Hirudinea has around 500 species. So they are present in the freshwater, some in the seawater, some are terrestrial, and we know we are going to read about the leeches soon. So the Hirudinians, they look like oligochaetes in most of the characteristics. Again, you can see that, there is a common ancestry. And if you see, they are mostly ectoparasites and blood-sucking, dorsal-ventrally flattened body. Some are carnivorous predators too. And number of segments in this body is usually specific, around 33 to 32 to 34 segments always. And each segment is again divided into number of annuli. And you have appendages, parapodia, and setae that are completely absent. Head is distinct but has eyes. And a clitellum is not present always, but it is present only during the breeding season. And this is present around the segments 9, 10, and 11. Body wall has a layer of or dermis. In addition to that, you have the circular and longitudinal layers of muscles. You also have oblique muscles in the class Hirudinea. And then they differ from the other annelids because they do not have coelom. So what happens is because there is an extensive growth of parenchymatous tissue, which is known as the botroidal tissue. So the coelom is reduced to small spaces or sinuses or channels. So example, in genus Hirudo or other allied leeches, you have botroidal tissue that is present inside the body. So this reduces the coelom. And digestive system is suitable because they have a parasitic life. It is in that manner itself, you only have a storage part and a digestive part. And the storage part is known as the crop. And the saliva has an anticoagulant. That means as soon as, something we get hurt, what happens is the blood starts oozing out. And then you get to see that after some time, the blood oozing or the bleeding will stop. That is because we have a lot of coagulating factors that are present inside the body. I do not know. There are almost around 13 to 15 factors. Hageman's factor, Christmas factor, then you have all these factors, which is the blood clotting factors present inside the body. So so what happens is Hirudinea or the leech releases an anticoagulant because of which the blood does not clot. This is known as the hirudin. And when you see, is there any doubt? Okay. So there are no special respiratory organs present when it comes to the respiratory system. Cutaneous respiration is present. And then when it comes to the excretory organs, you get to see that there are nephridia, around 10 to 17 pairs. And ammonia is the excretory product. And again, as in other annelids, same kind of nervous system, brain, nerve ring, and ventral double ganglionated nerve cord is present. And again, leeches, when it comes to their reproductive part, you have there again hermaphrodites, gonads, genital ducts are present in fixed positions. There is a sperm exchange that can take place between two leeches and is similar to that of the oligochaetes, and internal fertilization, and these fertilized eggs discharged into the cocoon. A clitellum develops again, like I told you, in the segments 9, 10, 11. During the breeding season, this forms the cocoon. Direct development to no larval stages. Example is Hirudinaria granulosa, which is the Indian cattle leech, and Pontopdella, which is the skate sucker, Branchelion, which is a marine leech. So when it comes to the fourth class, we get to see that there is archaeannelids. Again, they are a group of annelids. They have some resemblances with annelids. So they're primitive, and you can actually call them as ancestors to the annelids. So with the simple fact that whatever organs that are there in these particular annelids, they used to resemble the annelids. So there is a degeneration of the organ systems as compared to the annelids. That is why they are known as archaeannelids, archae primitive. So they are again treated as a separate minor phylum today. So when you get to see this class, they include around a dozen genera that are present. The small, mostly marine, and some of them are freshwater again. No particular segmentation is seen, and even if it is present, it is completely internal. And then no parapodia, so no locomotory organelles, no setae present, and head is completely distinct with certain sense organs. Prostomium, which is the oral part, which is present, oral segment is small, peristomium is large. The next, the segment after the prostomium is known as the peristomium. And then you have a circlet of adhesive papillae present around the anus in one particular species, that is the Polygodius. You have a ventral ciliated groove present in the Protodrilius. And again, epidermis is distinct. They might or might not have cilia, and other internal organs, even if they're present, they're all completely reduced. And then you get to see that you have transverse septa. Again, I told you internal segmentation is present, but not external segmentation. And so they have a spacious coelom as well. Circulatory system consists of a dorsal and a ventral vessel, which is starting from the anterior end and stopping at the posterior end. You have a nephridia present in each segment. Again, they may be closed with solenocytes, or you have nephrostomes that open into the coelom, and sexes are separate. Example is Polygodius. Protodrillus is like, monoecious, that is, it is a hermaphrodite. You have both male and the female organs in the same species, and there are no special gonoducts present. Development is indirect in certain animals such as Polygodius, direct in the case of Protodrillus, and larval form, which is Lowen's larva, is seen in Polygodius, and this is similar to that of the trochophore larva. So, in addition to these, you also have Archaeurida and Sipancula, Priapulida. All these are related to the annelids, because there is some sort of segmentation, metamerism, larval development, presence or absence of setae, coelomic cavities, the body wall construction, and all of these is there. That is kind of seen at some point of the other. So their systematic position is not exactly clear. So they have given you examples of pictures of this particular archaeannelids, that is the Polygodius and Protodilus. Now let me share again, and I'll start with the leeches now. Hmm. Not now, let Nanama watch. Hmm. wear your glasses. Right. So when it comes to leeches, they belong to the class Hirudinaria, class Hirudinea. Around 300 species of leeches are present, and they're found in freshwater, tanks, ponds, lakes, you name it, they are present in such places basically. They show great diversity in their habitat also. And the common Indian species are Hirudinaria granulosa, then the species Viridis, Hirudinea javanica, Hirudinea manilensis. And you also have tiger leech, that is Hemadipsa, lives in the South Indian hills basically. And Hirudinaria granulosa is the Indian cattle leech. So you have quite a number of important questions in this. So they'll ask you, they like, for example, one question as given in the at the back of your textbook, you have one important question, essay type, which is describe the structure of a typical nephridium of leech. That is one thing they have asked. And in short answers, they have asked what are the segmental receptors of leech. So you've got quite a number of questions that keep coming from this particular chapter. So if possible, please pay attention, and it will be easier for you to prepare for your exams again. So when you come to the classification of leeches, they belong to phylum Annelida, class Hirudinea, order Gnathodelida, genus Hirudinaria, species Granulosa. Again, like I told you, it lives in different places like ponds, lakes, tanks, and all fresh water. So again, it swims by vertical undulations or by a looping movement. And it is an ectoparasite on fishes, frogs, cattle, men also. That is, whenever you go and you step into some waters wherein there are leeches, it can hold you also. And they are sanguivorous, that is blood sucking. They feed on the blood of vertebrates. Excuse me. Right. So that's what a full grown specimen is, around 30 to 35 centimeters. Body is dorso-ventrally flattened. It looks like a strap shaped when there is a contraction of the body, the muscles basically. And when you get to see the color, the dorsal surface is olive green in color, or then the ventral surface is orange red, and you have distinct shapes, stripes of orange or yellow or black. And on the dorsal side you have a long median black stripe that is present. And again, you have a fixed number of segments, around 30 to 34, 32 to 34. And you always, there are around 33 segments present. So again, they have different kind of annuli in each segments, in fact. And the first and the segment have one annulus each. And third segment has two. Then fourth to sixth have around three annuli. Segment 7 to 22 have around five annuli each. Then 23 to 26 have around two annuli each. And around from 27 to 33, you have one annulus each. So that's how it is. Some very graphical pictures of the leeches, how they look basically. This is your tiger leech, Hemadipsa. This is Hirudo medicellanis. All these are sanguivorous leeches, sanguivorous, that is blood sucking. This is the Hirudinaria. So what happens is some segments with the segments which are having less than 5 annuli are known as incomplete segments basically, and with more than 5 annuli or with those with 5 annuli are called as the complete segments. So that is from 7 to 22, they are known as the complete segments. And again, each annulus is divided into small rectangular areas by longitudinal wrinkles. And each area as a sort has a sensory organ called as an annular receptor. And so around 18 such receptors are present on the dorsal and the ventral surfaces. And besides these, the first annulus of each segment bears a large sensory organ called as the sensory or the segmental receptor. So this is an annulus. As you can see this particular picture, you have the longitudinal wrinkles. These are the annular receptors. This is how it is. You have the prostomium, you have different eyes in different positions basically, as you can get to see. So same picture is there in your textbook also. So when it comes to these receptors or eyes, you see the picture, then you'll get an idea. So you have each rectangular area has an elevated sensory papilla, and that is known as the annular receptor. And there are 18 such annular receptors, both on the dorsal surface as well as on the ventral surface of each annulus. And there are large sensory papilla, that is the first annulus of each segment. You have around one large sensory papilla, 4 sensilla are on the dorsal side, 4 on the ventral side. And first annulus of each segment is known as the sensory annulus. And there are 5 pairs of eyes on the dorsal surface. First 2 pairs are on the 2 segments. First 2 segments. 3, 4, 5 are on the first annulus of the third, fourth, and fifth segments. So there is a particular number in which these eyes are also present. So the first annulus of the third, fourth, fifth segment has the eyes, third, fourth, fifth pairs of eyes. And so these the third, fourth, fifth segments are also known as the ocular segments. And the part of the body before the first ocular segment is called as the prostomium. And prostomium bears the prostomial glands and receptors of touch, sight. Therefore, it is a very highly sensitive area. So this is the prostomium. And you have suckers also present. So this is the anterior sucker, as you can see in the picture. So you have 2 different suckers, anterior and posterior. And anterior sucker is also known as the cephalic sucker, and or the oral sucker, because it has the mouth also in the same place. It is formed by the fusion of prostomium and a few anterior segments only on the ventral side. It is oval in shape, and there is a hollow cup-like preoral chamber that leads to the mouth. And this anterior sucker's function is adhesion and feeding. You also have a posterior sucker over here, and this is for the it is also known as the anal sucker, and it is present at the posterior end of the body, disc-like, highly muscular. It is formed by the complete fusion of the last 7 segments in the body, much larger than the anterior sucker. It is also ventrally directed, and this is only for adhesion. Anterior sucker is for both adhesion and feeding. Posterior sucker is only for adhesion. Now, when it comes to the divisions of the body, you get to see that the body is again divided into the cephalic region, the clitellar region, the preclitellar region, and the clitellar region. So what happens is, in the cephalic or the head region, you get to see that the first 5 segments, the prostomium, and then the anterior sucker, mouth, eyes, they're all present in the cephalic or the head region. But nephridopores are absent in this part. You do not need excretory organs in the cephalic or the head area. And then you have preclitellar region, which is present in the seventh segment 6, 7, and 8. So in this, you have nephridopores present. And then you have the clitellar region, which is complete 3 segments, 9, 10, and 11. And it is a highly glandular region. And there are nephridopores. You have both the male and the female genital apertures that are present in the clitellar region. And when it comes to the middle region, follow this picture, you'll be able to understand. Just keep following. So in this, again, in the middle region, starting from segments 12, to 22. You have, this is the largest region. Again, you have only the nephriospores present over here. And this 23 to 26 form the caudal region. And again, nephriospores are absent. This is the last segment has a dorsal anus. Then you have posterior sucker. Again, from 27 to 33, these segments are completely fused to form the posterior sucker, and intersegmental furrows are present and they are obscure. These 7 segments are arranged as concentric rings and they form a single annulus. Now, when you get to see the external apertures, you get to see that mouth is present as a tri-radiate with a preoral sucker, in fact, or a preoral chamber of the anterior sucker. Anus is again a mid-dorsal aperture at the base of the posterior sucker on the 26th segment. And then you have 17 pairs of nephriospores from segment 6 to 22. And you have a mid-ventral. So on the ventral region, on the middle of it, you have the male genital aperture between the annular groups 2 and 3, second and the third annuli of the 10th segment. And you have a thread-like penis which will be protruding out during the copulation time. And you also have a mid-ventral female genital aperture, which is an interannular groove. So between the second and the third annuli of the 11th segment, you have a female genital aperture. So again, when it comes to the body wall, you get to see that the body wall has cuticle epidermis, and you also have the muscular layer, and you have the botroidal tissue. So in this, again, the cuticle is outermost, thin, delicate. You have got a good picture of this in the, please follow this picture. Same picture is, in the colored form in your textbook, it is not colored. So cuticle is the outermost non-cellular, thin, transparent layer present, and it is periodically shed off after, especially after a heavy meal. And it is again produced with the help of the epidermal glands, and it has a protective function in nature. And beneath this, you have the epidermis. So epidermis is below the cuticle, has a single layer of columnar cells which are hammer or mallet shaped, and then this gives the epidermal cells a firm grip so that it can hold the vertebrate onto which it is sucking the blood from, and so that it can become elastic or ribbon like, especially during locomotion or especially while swimming. So the interspaces between the inner ends of the epidermal cells, you have the fibrous connective tissue present. So this you also have the pigment cells and the hemocoelomic cavities present. You can get to see here the fibrous connective tissues, the hemocoelomic cavities or capillaries present. And this hemocoelomic capillaries help it for the efficient respiratory surface basically. And some of the epidermal cells are modified into the unicellular epidermal glands. These epidermal glands only produce the cuticle, and you also have multicellular receptors. So epidermal glands are pear-shaped or tubular, and the function, based on the position and the function, they're again classified as slime glands, sucker glands. You can get to see them pear-shaped glands basically. The slime glands or sucker glands, prostomial glands, and Glytelar glands. And according to the name given, they produce that kind of thing. For example, the sucker glands help, in groups. They're present in groups on both the suckers, and their suction helps to attach the body to the substratum during locomotion. The prostomial glands are found on the prostomium, have a distinct, and they're present as distinct series or groups. Their secretion forms two polar plugs that close the anterior and the posterior poles of the egg case. You'll get to know about it later. And when it comes to the Glytelar glands, you get to see that they're present in segments 9, 10, and 11. They become active during the breeding season. And there are two types, that is the chytogenous glands that form the wall of the cocoon, and the albumin glands that secrete the albumin. which is the reserve food in the cocoon, especially that is necessary for the developing embryos. You also have epidermal receptors, which are described under the sense organs, and then you have the dermis. So below the epidermis, you have the dermis basically, and it has a fibrous connective tissue cells, pigment cells, fat cells, etc. And the basal portions of the slime glands can also be seen in the dermis part. So when it comes to the musculature, we already know that we have the leeches have longitudinal, circular. At the same time, they also have the oblique muscles, which are present between the circular and the longitudinal muscles. They are directed spirally around the body like a coil. You also have dorsoventral muscles between the in each segments. In fact, these muscles run from the dorsal to the ventral side by the side of the alimentary canal. And you also have radial muscles. These muscles radiate from the alimentary canal to the body wall. They help, they are in the form of septa. And in addition to this, you also have erector and depressor muscles in the suckers that help in attachment and detachment of the suckers to any substratum. And this elaborate muscle arrangement is an adaptation to the different kinds of locomotion it needs to perform. You also have the botroidal tissue, which is a mesenchyme present between the body wall and the alimentary canal, and it is completely made up of pigments and richly vascularized cells, large, that are arranged end to end. They have a dark brown pigment having intercellular branching capillaries that are filled with fluids, and its function is probably excretory. That is, it almost completely fills the body cavity. Again, the botroidal tissues of two types of cells that are present, the fat cells and the yellow cells. So fat cells contain fat droplets and some glycogen. Yellow cells are filled with yellow, brown, or green granules. So excretory function. And then that's about it regarding the botroidal tissue. Or then when it comes to the functions of the body wall, again, protection of the internal organs and evaporation. To prevent evaporation, you have the cuticle, and you have the slimy coat that is present because of which it can escape enemies from holding onto it, richly supplied with hemocoelomic cavities, so they are respiratory in function. And quite a number of sensory receptors are present, because of which even a good sensory uh, function is seen over here in the leeches. And then the gland cells present in the walls secrete the plugs of the utica. We have already seen that it is the posterior prostomial glands. In fact, the egg case is also known as the utica. And then you have the gland cells in the Glytelar segments that secrete the utica and the what do you call it? The albumin. So albumin being the food for development of the embryos, in fact, for the developing embryo. So this is how locomotion is seen in Annelida. So you have the creeping, looping, or crawling and swimming kinds of locomotion present. All the different kinds of muscles help in this particular locomotion. In fact, so what happens is at the same contraction and extension is seen at the same time. So movement occurs on any kind of substratum similar to that of the looping movement of caterpillar. Posterior sucker is firmly attached to the substratum by the secretion of the sucker glands. Then the circular muscles contract, longitudinal muscles relax. So there is an extension of the body. Then what happens is anterior sucker comes and attaches itself to the substratum and shortens, because of which, and then the posterior muscles or the posterior sucker gets released from the substratum. And then again after this process is repeated. So this kind of movement is actually known as the leech-like locomotion. And when it comes to swimming, you get to see that the dorsoventral muscles and the get to contract. So you have undulatory waves that are being seen over here, and then the body moves from the anterior to the posterior end because of the contraction of the longitudinal muscles now. So this is all about the swimming or the locomotion. When it comes to the digestive system of Hirudinea, you get to see that the digestive system is composed of or divided of three parts: stomatium, which has the pre-oral chamber, and you can get to see in this picture, in this diagram. In fact, you have the pre-oral chamber, you have the mouth, you have the buccal cavity, and the pharynx, and the mesenteron, which includes the esophagus, crop. So this is the stomodium, this is the mesenteron, and this is the proctadium over here, which comes like, so you have the proctodium over here. So stomodium, mesenteron, and proctodium or proctadium. Yes. So the mesenteron includes the esophagus, crop, stomach, and intestine. And this is the proctadium, including the rectum and the anus. Now, leech is a sanguivorous or a blood-sucking creature. Again, blood is not readily available all the times. So when it is available only, the blood, it is going to suck in the maximum amount of time, and it is going to be stored. So you have a storage cavity, and that, the blood contains the digested foods, needs a little digestion. So the digestive function of the whole organism is completely or greatly reduced. So you get to see that this preoral chamber over here, and mouth, buccal cavity, pharynx, esophagus, crop, stomach, intestine, rectum, and anus is the digestive system. And based on it, again, you have different kinds of zones in the alimentary canal itself. This is the blood sucking, this is the blood carrying, storage, digestive and absorptive, and the egestion zones. So when it comes to the blood sucking zone, you get to see the preoral chamber and the mouth, the buccal cavity, and the pharynx. So till here you have the blood sucking forms. So what happens is, this cephalic sucker is applied onto the body of the host, skin gets punctured, blood oozes out, and again at this particular point of time, release of the anticoagulant takes place, and with which, it starts sucking at the same time. Mouth is a triradiate opening, you can see over here. And one ray of the mouth is the median dorsal, and the other two are ventral lateral. And you have a membranous velum. This is a membranous velum which guards the mouth, being present between the mouth and the buccal cavity. And it has three lips or gaps, out of which two are dorsolateral, one is median ventral in position. And this is the buccal cavity present over here. And it is adapted for puncturing the skin of the host, basically. And you have a cuticle over here. We're having minute teeth or denticles in a single row, giving the giving the edge a serrated appearance. And out of the three jaws, you have median dorsal and the other two ventral lateral, corresponding to the position of the rays of the mouth, so that they can get easily extended out. So around 42 to 45 button-shaped papillae called as salivary papillae are present. You see the salivary papillae over here. These are the ones. And the jaws are having three types of muscles: diagonal muscles, peripheral muscles, and longitudinal muscles. And the jaws protrude out through the mouth and cause a Y-shaped wound on the host body. Buccal cavity opens into the pharynx. The pharynx is a very highly muscular oval sac, has around 5 to 8 segments. Internally, the wall of the pharynx is in the middle region. It has around 6 to 12 folds or ridges. And you have the salivary glands surrounding the pharynx. And this is used as a suction pump for sucking the blood, and this opens into the esophagus. So over here in esophagus, you have the zone that carries the blood to the different kinds of storage organs. So esophagus is very narrow. It present on the 8th segment. Passage for blood from pharynx into the storage organs. Zone of storage, as you can already see, this is the largest part of the alimentary canal, which has the crop. See, this is the crop over here. These are the crop chambers. This is the crop. Extends from around 9th to 18th segment. Thin-walled, occupies around 2/3 of the visceral spaces. And then what happens is, you have the anterior part of each chamber, which is small. Posterior part is broad. and it has a large lateral diverticula also present, and they are also known as CK, and they are the storage pockets. So the size of the chambers slowly increases as you go to the posterior end. And so the last chamber is the biggest with the largest ceca, which is in the 22nd segment or even further backwards. And they again give rise to secondary ceca, which can be seen when the crop is extended. This thin-walled crop chamber ceca is used as an adaptation so that whenever there is availability of blood, it can completely store. So this is what we are talking about. So the crop ends in a funnel-shaped tube. Opening into stomach is guarded with a sphincter. So this is the first time we are listening to the word called as sphincter, which is a group of muscles which help in the control, that is the contraction or a relaxation of that particular muscles. For example, you have the pyloric sphincter, gastric, and then you have when it comes to the digestive system, the rectum. For example, you know that it has a sphincter, the anal sphincter. Contraction and relaxation of the anal sphincter helps in the release of the feces out of the body in vertebrates. So sphincter is a muscle which is used for contraction and relaxation. So normally it is contracted. When it is relaxed, it has a different purpose altogether. So when it comes to the fourth zone, or the zone of digestion and absorption, it is a very short one over here because there is no great need for digestion. Already digested food is available. So you have the stomach and the intestine. You can see that in the picture. And stomach is a small heart-shaped part occupying the 19th segment. Inner lining has a lot of transverse folds which are anastomosing with one another, and that they're joining one another. Stomach has secretory and absorptive epithelial cells. Intestine is not very clearly demarcated externally from the stomach. Thin-walled, narrow. From the 19th segment to the 22nd segment, you have the intestine. And again, it has longitudinal transverse folds which look like villi, and this increases the absorptive surface, and intestine opens into the egestion zone. So egestion zone has the rectum and the anus. So rectum is thin-walled and slightly dilated. Posteriorly, it turns upwards. No digestion occurs in the rectum. It helps in water conservation by reabsorption from that is the undigested food, and this opens outside through the anus. Again, it is the exit for the undigested food, present mid-dorsally on the 26th segment at the base of the anal sucker. So when you're drawing the diagram of this, when they ask you for digestive system, all you have got to do is draw the diagram and please label the parts according to the segments. You will not make a mistake. And then the same thing you write as points, you will definitely get all the marks. Sorry. Yes, right. So when it comes to the different kinds of digestive glands, you have salivary glands, you have isolated gland cells, and then you also have the gland cells. This gland cells secrete the anticoagulant called as a hirudin that helps in blood, prevention of blood clot at the site of the wound, so that the leech can continuously suck blood. So when it comes to feeding, digestion, and absorption, you get to see that it feeds on cattle and other domestic animals that enter the water. So it holds by the posterior sucker, adhesion by posterior sucker, and oral sucker is for to penetrate the skin so as to start sucking the blood, and it painlessly, because of the presence of the anticoagulant, also painlessly punctures the skin of the host. Then the functions of, or with the help of the preoral, the oral cavity, the buccal cavity, pharynx, oozing blood is sucked in. And then what happens is the radial muscles around the pharynx contract. So what happens is this increases the pharyngeal cavity volume, because of which pharynx acts as a suction pump. So if it weighs around 5 grams, it can suck blood up to 5 to 10 times more than its own weight. I'm just quoting over here. Please don't write 5 grams. It can weigh more or less for that matter. So it can suck blood which is 5 to 10 grams, 5 to 10 times more than its own weight. And this will help it survive for more than quite a number of months to almost a year, in fact. And the hirudin from the salivary secretion prevents the blood from clotting during the oozing. And from the pharynx, the blood reaches the crop. Then in the crop, the red blood corpuscles are hemolyzed. That is, RBCs are broken down. Spleen is where RBCs are broken down in the vertebrates. Over here, the crop, the RBCs are hemolyzed, that is broken down, and hemoglobin in the RBCs goes into solution. Blood becomes dark red. So this is again passed on to the stomach, drop by drop through the sphincter aperture, and in the blood, in the stomach, the blood is digested. So proper mechanism of digestion is not yet given properly, but also a couple of scientists, Abderhalder and Hayes, found a pectolytic enzyme that digests protein in the alkaline medium. And Busing, other scientists, reported that digestion is due to bacteria present in the gut of the leeches. And so hemoglobin of the ingested food is directly absorbed by the cells of the stomach. Globin from the blood. Globin is the protein part, mainly used as food. Absorption occurs in the intestine. Takes more than a year for the leech to completely digest and absorb one full meal. Very little undigested food is sent out from the rectum through the anus. Digested food is in the form of glycogen found abundantly in the cells of the stomach. So this is about the alimentary canal. So again, you get to see that, there is a coelom and a coelomic system, in fact, present, which is present. So coelom is different from that of the other annelids. You have the botroidal tissue occupying a lot of area in the in the earthworms itself, and there are 4 more longitudinal channels with different kinds of branches, and all these are lined in the coelomic epithelium. And you have the coelomic fluid with dissolved hemoglobin in it, and this fluid is not real blood but a combination of coelomic fluid and blood. And these channels are known as the coelomic channels, hemocoelomic channels. Fluid is the hemocoelomic fluid. Spaces in the testis sacs and ovi sacs and around the vasa differentia are the true coelomic sinuses of the organism, as they are lined with the coelomic epithelium, and that this produces the gonads. And then you have this is filled with colorless coelomic fluid around this gonadic testis sacs and ovi sacs. This suggests that sinuses are the original remnants of the coelom. They get separated before the hemoglobin development in the blood, basically. So when it comes to the hemocoelomic system of hirudin area, you get to see that true blood vessels are not exactly present, and this whole function is taken by with a system which has the hemocoelomic fluid and hemocoelomic channels with their branches, and hearts are absent. So it is a modified combination of coelom and the circulatory systems. There is no heart present in the leeches except as a and the walls of the lateral channels are pulsating, rhythmically contracting. This causes the circulation of the hemocoelomic fluid. And you have adrenaline, which is helping in the acceleration of the contractions that is released from the ventral nerve cord. They're also under the influence of inhibitor and accelerator nerves from ventral ganglia. So again, the hemocoelomic fluid has dissolved hemoglobin, different hemocoelomic channels, branches, capillaries, colorless amoeboid corpuscles as well. And these include dorsal, ventral, and 2 lateral channels. This is given in the pictures, basically dorsal, ventral, and 2 lateral channels. And you get to see that the dorsal channels have branches also. And in this, the walls are also not present in the dorsal channels. Hemocoelomic fluid flows posterior to anterior in this channel. Anteriorly in the 6th segment, it breaks up, forms a network above the alimentary canal Posteriorly, in the 22nd segment, it is bifurcated into two. So again, this dorsal channel is a distributing channel, and it again gives off to dorsolateral and dorso-intestinal vessels. Now, ventral channel, when you come to see, again, you get to see that this ventral channel is mid-ventral, below the alimentary canal, running from the anterior end to the posterior end. They are not exactly contractile, that is, they do not contract, they are thin walls. Valves are absent in this channel, and hemocoeloic fluid flows from the anterior to posterior end. And it is much wider than the dorsal channel, encloses the peripharyngeal cavities, subpharyngeal cavities, peripharyngeal connectives, subpharyngeal connectives, subpharyngeal ganglionic mass. It also has the ventral nerve ganglion, ventral nerve cord, terminal ganglionic mass as well. It is a distributing channel, and it gives off the branches to various organs in the segment and to the nephridia as well. So you have the first pair or cutaneous branches and second pair or the nephridial branches. So this is how it is. And again, if you get to see, the first pair or cutaneous branches are subdivided into two different branches, the ventral branch and the abdomino dorsal branches, in fact. And then you have a second pair of nephridial branches. These are again, you have around a segment of a pair of these branches come from the level of the segmental ganglion, and on each side you have the nephridial branch running outwards, and onto the testis sac of its side enlarges into 2 or 3 closely set sacs called as the perinephrostomal ampullae that enclose the ciliated organ of the nephridium. It continues to run outside, supplies the hemocoelomic fluid to the nephridia and the body wall. Now, there are 11 pairs of nephridial branches that occur from the 12th to the 22nd segment, in which the testes are also present. I will have to request you that I'll at least, finish the leeches, and later the excretory system will be dealt in the next class. So you please bear with me till at least 5:30. Let me finish the leech and then I will relieve you. So this is about the ventral channel and its branches, as you can see. So you also have the lateral channels. In the lateral channels, you have one on each side of the alimentary canal. You have thick contractile walls also present. So in this, you have the blood flow from the posterior to the anterior end in these channels, and the two lateral channels anteriorly break up into capillaries in the 6th segment. Posteriorly, they open into the dilatation of the ventral channel again. Now they are both distributing and collecting in nature. Each segment receives two branches. You have lateral and laterodorsal. So lateral and laterodorsal. These are the laterals. These are the laterodorsals. And ventral, these are the two collecting branches. And also in each segment, you have the ventral, which is a distributing branch. So when it comes to the lateral, these are the laterals present. So what happens is, this collects the hemocoelomic fluid from the body wall and the nephridium of its side, joins the lateral channel at the level of each nephridial vesicle. You also have the laterodorsal branches. So you see, these are the laterodorsals, as you can see over here. This is formed by the branches from dorsal and the dorsolaterals, regions of the body wall, gut wall, and nephridium. This helps, joins the lateral channel with the help of two commissures, which is the dorsal commissures of the lateral channels. These are the dorsal commissures of the lateral channels. And also you have the lateral commissures. Right. So in the lateral commissures, each segment, the dorsolateral, that is this dorsolateral, is connected to by a transverse loop above the dorsal channel. These loops are called as the dorsal commissures of the lateral channels. And there are 17 such commissures from the 6th segment till the 22nd segment. And the lateral commissures, each segment, the lateral dorsal is connected by the laterolateral over here. See, these are the lateroventrals, right? So you have the laterolateral commissures over here that connect. You have got a better picture over here if you see. The laterodorsal is connected to the laterolateral of its side by a longitudinal commissure, which is known as the laterocommissure. This laterolateral, laterodorsal branches, they're all laterolateral and laterodorsal. All of these are collecting channels, basically. They collect the blood. Lateroventral branches are also present. These are the lateroventral branches that are present. And you get to see that their function is again, this gives off a branch to the nephridium and the ventrolateral regions of the body wall. So what happens is you again have an anterior and a posterior branch in each segment again. So these form again a ventrolateral commissures. From the 6th segment to the 23rd segment, there are 18 such rhomboids. So this shape is like a rhombus shape. So the rhomboids or ventral commissures, and this is present one in each segment. So this supply hemocoelomic fluid to the alimentary canal and the reproductive organs. So the branches of the lateral channel supply the hemocoelomic fluid to the skin, nephridia, reproductive organs, and the alimentary canal. Branches of the lateral channel, they again, lateral and laterodorsal collect the hemocoelomic fluid from the upper and the lower body wall, alimentary canal, nephridia, and reproductive organs. So this is how it is. And when it comes to the capillary system, in the leeches, you get to see that you have botroidal capillaries, intermediate capillaries or intramuscular capillaries, and superficial capillaries or cutaneous capillaries. All of these, again, the botroidal capillaries receive the hemocoelomic fluid from the ventral channel and join the lateral channels. Intermediate capillaries again receive supply from the lateral channels. Then they again give it to the botroidal capillaries. And you also have superficial capillaries which go into the epidermis and communicates with all the different 4 channels that are present. So this is how the course of circulation is. You can get to see that in the case of what do you call it, the leech for that matter. So there is a definite direction in which the blood goes in the leeches. So in the dorsal, and you please follow this particular picture, you can get to see that in the dorsal and the 2 lateral channels, it flows from posterior to anterior end. In the ventral channel, it flows from the anterior to posterior end. Posteriorly, in the 26th segment, all the 4 channels directly communicate with each other. Dorsal and ventral channels are distributing in nature, and the lateral channels are both like distributing and collecting channels. Dorsolateral body wall of the gut received through the dorsal channel. From this, the fluids are collected by the dorsolaterals, carried to the lateral channels, and from the mid-dorsolateral, ventral parts of the body wall, nephridia, receive the fluid from the ventral channel, collected from these parts by the laterolaterals and the laterodorsal branches of the lateral channel. So the lateral channel connects from the laterolateral and the laterodorsal branches. Now the lateral channel through the lateroventral branches supplies the fluid to the nephridia, reproductive organs, floor of the gut, ventral body wall, laterolateral and laterodorsal again carry these to the lateral channels present. So this is how it is. You can see it is completely definitive. That means that there is only blood flow in this particular direction. Please just give me 1 minute. So this capillary system of the hemocoelomic channels and commissures, all this is also quite important circulation. Please just have a look at it. And when it comes to the respiratory system, again, there are no specific respiratory organs. Gaseous exchange takes place through the permeable, what do you call it, skin, in fact, and hemocoelomic fluid only. This has this extensive capillary system, capillary loops. So exchange of gases takes place through this. And since they live in water, damp soil, etc., skin is always kept wet by surrounding water. Mucus is also secreted so as to prevent the drying. When it comes to the excretory system of Hirudinea, you get to see that it is segmentally arranged. There are around 17 pairs of nephridia. This is the picture, as you can see. Again, this is an important question asked. So you have around 17 pairs of nephridia present in segments from 6th to 22nd, and first 6 pairs are present on segments 6 to 17. They are known as the pre-testicular segments, 6th to 11th. And then you have the remaining 11 pairs in the testicular segments, that is from the 12th to the 22nd. And nephridia lying in the pre-testicular segments and the testicular segments are known as the pre-testicular and the testicular nephridia, respectively. Now, a testicular nephridium picture is also given. It is a horseshoe-shaped structure. We'll just kind of show you. This is how it is. This is a typical nephridium. Again, there are 11 pairs of testicular nephridia, and it has initial lobe. This is the initial lobe. You have an apical lobe. This is the apical lobe. You have the main lobe over here, and then you have the inner lobe. This is the inner lobe that is present. And you have a vesicle and a vesicle duct and ciliated organ. So initial lobe is also known as the testicular lobe, basically. And this is a long, narrow, transparent cord-like structure made up of a single row of elongated hollow cells with intracellular canal that gives off diverticula. So you have diverticula also coming off from this intracellular canals, basically, as you can see over here. And it closely coils around the apical lobe. Inwardly, it ends blindly close to the perinephrostomal ampulla of its side. Initial lobe posteriorly joins the apical limb of the main lobe. Now you have the apical lobe over here. As you can see, this is the apical lobe. This lobe is stout, lies antero-posteriorly beneath the crop. Anterior end of this lobe is slightly bent like that of a walking stick. Cells are large, have a lot of intracellular canals. And this is the main lobe, as you can see over here. This main lobe again has anterior and posterior limbs. Anterior limb is long, posterior limb is short. Again, it has a ventrolateral between two diverticula of the crop, basically. And when it comes to the inner lobe, this is the inner lobe, as you can see. This inner lobe is narrow, lies throughout the concavity of the main lobe. Inside the main lobe, it is present. And this runs forward along the outer border of the apical lobe for about half of its length, till here, of the apical lobe. Cells are again long, tubular. Vesicle and vesicle duct. Vesicle stores the excretory fluid. Vesicle duct is narrow, arises from the inner ventral end of the anterior limb of the main lobe. So again, at the junction of the vesicle and the excretory duct, you have a sphincter which controls the excretory fluid that is being released out, or the outflow of it, basically. You also have a ciliated organ. As you can see, the ciliated organ, there are around 11 pairs associated with the testicular nephridia. And this lies in the dilatation of the hemocoelomic system. These are also known as the perinephrostomal ampulla. It is suspended in the ampullae by around 4 to 5 strands or trabeculae that arise from the walls of the ampulla. The ciliated organ consists of a central spongy reservoir, as you can see, and ciliated funnels. So this has connective tissue cells in the middle and this manufactures the coelomic amoeboid corpuscles. Reservoir is perforated by innumerable number of pores. So many number of pores are present, and on its outer and inner surfaces, it is covered with outwardly directed cilia as well. You can see the cilia over here, right? So in the embryo, the ciliated organs are connected to the nephridium, but in the adult, this connection is basically lost when they become a part of the hemocoelomic system. You also have the pre-testicular nephridia from segment 6 to 11. This is the ciliated organ. This is a funnel with the cilia present outward, as you can see over here. So this is the ciliated organ enclosed with the perinephrostomal ampullae. We have already seen all of this. So when it comes to the pre-testicular nephridia, from 6 to 11 segments, you have this pre-testicular nephridia. And because of this, they do not have testis-sacs, perinephrostomal ampullae, and ciliated organs. And so the initial lobes of these nephridia lie loosely inside the general connective tissue as such. So when it comes to the physiology of excretion, you get to see that it is, you have a lot of hemocoelomic system, which is, completely or richly branched. Then they separate the waste material, and then this is finally carried to the vesicle through the intracellular canal, central canal. From there, it is excreted out through the nephriospores. And this nitrogenous waste in the leach is mainly ammonia. Also contains little quantities of urea, purines, creatinine. So this leach is basically ammonotelic in nature. When it comes to the nephridia also act as osmoregulatory organs. So that means the osmotic fluid, the regulation of the balance of it is maintained by the nephridia, because water as such continuously enters the body of the leech as they are present in water. So excess of water is removed by the nephridia so that the constant balance of the body fluids is maintained. When it comes to the nervous system of hydrogenaria, you get to see that there are three parts: the central nervous system, peripheral nervous system, and the sympathetic nervous system. Again, the central nervous system has a pair of cerebral ganglia and a pair of pharyngeal connectives, a pair of subpharyngeal ganglionic mass, ventral nerve cord, terminal ganglionic mass. Let me tell you again, I will just finish the leeches and then you will be relieved. I have the reproductive system also present. So another 5 minutes and I can finish the class. So please bear with me. So when it comes to the terminal, this is the central nervous system that is present, and it is enclosed, this terminal ganglionic mass. The entire central nervous system is enclosed with within the ventral hemocoelomic channel. A pair of fused cerebral ganglia forms a small brain that is present above the pharynx and the fifth segment. Brain is shifted because of the reduction or in the size of the prostomium. Prostomium is small, so because of that, even the brain is small. From the brain, you have a peripharyngeal connective that is present. This is the picture that you can see. And these join the subpharyngeal ganglionic mass below the pharynx and the fifth segment. This is formed by the fusion of pairs of ganglionic mass. Go to Nanama. Chalo. You have a double ventral nerve cord that is covered, and this is covered by a protective sheath called a neurolemma, something like our the neurolemma that we have. This runs along the midventral line from the subpharyngeal ganglionic mass to the terminal ganglionic mass lying within the posterior sucker. So around 21 double ganglia are present from the first annulus onwards, which is known as the somite, from the 6th to the 26th segment again. This is a large ovoid body, that is the terminal ganglionic mass. Again, there is a fusion of 7 pairs of ganglia over there, as you can see, in the case of the posterior sucker, like in the same manner. When it comes to the peripheral nervous system, you have a pair of nerves that are, given out throughout the body. So you have nerves given off by the cerebral ganglia, subpharyngeal ganglionic mass, ganglia of the ventral nerve cord, terminal ganglionic mass. All of these again give the nerves or they constitute the peripheral nervous system. These nerves travel to different parts of the body. Cerebral ganglia give nerves that run anteriorly, supply the branches to the nervous system for the eyes, prostomium, roof of buccal cavity, and laterally that is laterally from the subpharyngeal ganglionic mass arise 4 pairs of nerves. They reach the second, third, fourth pairs of eyes. Again, subpharyngeal ganglionic mass gives nerves to the roof of the buccal cavity, muscles of the body wall, segmental receptors in the first 5 segments. From each ganglion of the ventral nerve cord, 2 pairs of the nerves arise. They give rise to the anterior laterals and the posterior laterals. Anterior laterals come from the anterior part, posterior from the posterior part of the posterior part of the ganglion of the ventral nerve cord. Terminal ganglionic mass supplies the nerves to the posterior sucker. So when it comes to the sympathetic nervous system, you get to see that again a lot of nerve plexus, means junction of nerves are present, and these are all autonomic, and it is present beneath the epidermis, inside the musculature of the body wall basically, and between the alimentary canal as well. So the nerves in the system join certain cells on both the sides, that is the peripharyngeal connectives. And again, they also join certain multipolar ganglion cells of the plexus above the alimentary canals. Now, the sense organs of area, you have around 4 different types of receptors: free nerve endings, annular receptors, segmental receptors, and eyes. You can get to see them in the picture. And then, the free nerve endings are similar to chemoreceptors. Annular receptors are for 18 receptors that are present in the middle of each annulus. Segmental receptors are small. There are also tactile or tango receptors or touch receptors. Eyes, again, they help in perceiving light from darkness. This is about their basic function. And when it comes to the reproductive system, this is the last part. Another 5 minutes, your class will get over. Please bear with me. So the male leeches are hermaphrodite, that is both male and female reproductive organs are in the same organism. Self-fertilization does not occur. During copulation between the two worms, you have exchange of spermatozoa taking place, and all leeches are protandous, that is the male reproductive organs develop beforehand itself as compared to the females. The timing is different. So in the male reproductive organs, you have the testis sac, vasa differentia, vasa efferentia, epididymis, ejaculatory ducts, and atrium. Around usually 11 pairs of testis sacs are present, one in each segment from the 12th to 22nd. And they have each testis sac also has a spherical coelomic sac. So they form the spermatogonia or the sperm mother cells that get budded off, and then the spermatogonia float into the coelomic fluid within the testis sac. They develop into the spermatozoa. Now, vasa efferentia from the posterior border of the let me show you the picture, it is easier. This is how it is. So vasa efferentia is one wherein you have arising from the posterior border of the testis sac, connects the testis sac to the vas deferens. Vas deferens is from the 22nd to the 11th segment, goes from in the ventral body wall. It they enlarge, form compact masses called as the epididymis or the sperm vesicles, and then they they form the short ejaculatory duct or the ductus ejaculatorius. This leads to the atrium. Atrium is a pyriform sac that is present, and this is present in the 9th segment over here, as you can see. And then it consists of 2 parts, the prostrate and the penis sac. Prostrate is the anterior part, and these have glands open into the central prostrate chamber through the ducts. Penis sac is present, that is elongated muscular chamber. It is eversible. It has a filamentous coiled tube-like penis, which is eversible. It protrudes out mid-ventrally between the 2nd and the 3rd annuli. And from the testis sac, spermatozoa are carried to the vas deferens through the vas efferentia. From here, they reach the epididymis. And from the epididymis, the spermatozoa are carried to the prostratic chamber. From there, they form the spermatophores by prostratic secretion. These again enter the canal into the penis, and then they are transferred to the vagina during copulation. So when it comes to the female reproductive system, you have ovaries, ovisacs, ovaries, oviducts, and a common oviduct and the vagina. So a pair of ovisacs, one on either side of the ventral nerve cord, are present in the 11th segment. They contain ovary and ova budded off from the ends of the ovaries. Oviducts are very short, slender pairs of tubes that are present basically. This arises from the 11th segment onwards. The two oviducts unite. They form a common oviduct. This common oviduct then, let me show you. Yes, please see this picture. You will get the idea. This common oviduct, again, you have a small number of albumin glands that are present, and this ductule arises from each gland and they open into the common oviduct. And the posterior part of the common oviduct is like an S shape, and this opens into the vagina. These are the ovisacs. This is the ovary. This is the vagina over here. And it is a pear-shaped organ in the posterior pair of the segment 11. And it has circular muscles and a cuticle. Increases in size during the breeding season. Opens into the duct through which the female genital pore opens in the ventrally, mid-ventrally, between the second and the third annuli of the 11th segment. For the male, it is the 10th segment. For the female, it is the 11th segment. So segmental differentiation is present. Ova from ovisacs enter the vagina through oviducts and the common oviduct. Here they are stored for some time. Vagina is a receptacle for spermatophores, and then it received from the another leech as such. Fertilization, cross-fertilization takes place. Breeding season is between March and April. During copulation, it can be either in land or in water. Two leeches meet head to tail position so that male genital aperture of one is against the female genital aperture of another. And then the penis, and with the exchange of the seminal fluid with the spermatophores, it enters into the vagina of the other leech. Copulation takes place for about an hour, and then the leeches separate. Fertilization occurs inside the vagina, hence it is known as an internal fertilization. Now, cocoon formation and cocoon. This is how copulation takes place. And when it comes to the cocoon formation, the cocoons are deposited in 2 minutes more. We are done with the class. Cocoons are deposited in April, May, June, and a temporary clitellum is formed. You can see over here. For around the 9, 10, 11 segments during the breeding season, the Glytelar glands secrete several layers of broth which appear as snow-white girdle around the clitellum. This is secreted into the girdle by the Glytelar glands, and then fertilized ova are discharged into it through the female genital aperture, and from there it withdraws into the preglytelar region from the girdle by rhythmic movements of the prostomium when it reaches the anterior pole of the cocoon. It secretes another plug over here. This process of formation of the plug takes around 1 hour. After this, it comes out, but it keeps the anterior end in touch or in contact with the posterior pole of the cocoon. This is the anterior pole, posterior pole of the cocoon. It is still in touch with it. And then after another hour of completion, finally the leech leaves the cocoon. So cocoon formation finally takes around 6 hours. So the froth on exposure to air hardens, forms an outer spongy inner membranous layers, and it is light yellow or amber colored, barrel-shaped structure measuring about 25 to 30 millimeters in length and 12 to 15 millimeters in diameter. Chitinous wall of the cocoon has an outer thick spongy layer that forms a protective cushion for the cocoons. So that they are inside the pond, they lay in the clay or in holes, or in small holes that are present in the pond. Inner layer is thick and membranous, and a number of air bubbles are present so that the cocoon is kept light, floating a little bit. Then two ends of the cocoon are narrow, and each is closed by a polar plug. And inside the cocoon, you have this albuminous jelly fluid, which is the food for the developing embryo. Fertilized ova float in this particular fluid. And when the young leeches come out, this fluid is completely used up. Cocoons are deposited in moist places by the side of the water, but never inside the water. Development is also taking place inside the cocoon. Each cocoon contains around 1 to 24 embryos. Cleavage is unequal. Development is direct. There is no larval stage. In about 15 days, young leeches hatch after the polar plugs drop out. It may take around 5 years to reach a full-grown size for the leeches, and young ones are predaceous in nature, but only after, but they can mature only after feeding on mammalian blood. So now you have parasitic adaptations that are present. So it leads an ectoparasitic life. So they feed on the blood of cattle and man. So it is present in the shallow waters where it can easily reach the person. And when it comes to the segmentation, the segmentation helps in the easy bending of the body. Suckers, anterior and posterior, are present. Anterior for sucking or for the blood, posterior for attachment, and jaws are present so as to make a wound on the body of the host, so that when the blood oozes out, the leech can suck it. Again, you have a suctorial pharynx, which acts as a suction pump. Hirudin is the anticoagulant that helps in preventing the clotting of the blood, so that as much as possible blood is sucked into the leech. And then there is a multi-chambered crop that is present, having a very good function as such, and with its lateral ceca acts as a reservoir for storage of food. And it has a very slow digestive system, wherein digestion and absorption takes place very slowly, around 10 to 14 months in an adult leech. So it uses its food very, very precious, in a precious manner, very judiciously, and it makes complete use of the food maximum possible. And hermaphroditism, that is, in the unisexual animals only the female lay eggs, but in bisexual animals such as leech, every animal lays eggs, and this results in 100% production as against only 50% production in the unisexual animals. This gives a chance for rapid increase in their numbers or propagation. Cocoon formation helps in protection and developing embryos, so that you have more amount of survival chances. So, and hirudin, it is an anticoagulant which is actually used in medicinal purposes also. We take hirudin as medicine, so that they are like blood thinners and anti-coagulants, anti-blood clotting factors. So this is all about the hirudinaria, as you can see. Next class, I will try to finish the rest of the what do you call it? The annelida portion. In fact, please give me a moment. I hope you have all understood this so far.
Madam, can you send me PPT?
Sure, I was about to ask in case you okay. Acha, since the what do you call it? The host, I mean, it is not being live streamed right now. Do you want me to continue with the class, or you want me to stop it right here? Should I finish Annelida today?
Continue, madam.
Yes, no problem with you all.
No, madam.
All right, thank you so much. Just give me one more minute.
Madam, after the completion of class, can you send me PPT?
Sure, I will do that. But then, the host is not there. So in case they ask me, let me see that the university people are not there. So if at all I start finishing the class, is it okay? So you want me to finish the class?
Yes, ma'am.
All right, then I'll do that. If we finish
Yeah,
this Annelida today, at least, Arthropoda, because, the topics are very big and we don't have much time. I have only 2 more classes and you've got around 4 topics left. Impossible for me to finish. So I've got to take your time, unfortunately. So this is all about hirudinaria. So what I'll do is I'm going to stop share and I'll add one more PPT for that matter. So when it comes to the economic importance of earthworms, you know that, they are used for they help in fertilization basically. And they help in the breakdown of organic debris that is present, that is all the organic waste material. And so when this happens, they help in biodegradation of the waste. So they are known as the nature's gift for utilization of organic wastes. So they are bioreactors, excellent bioreactors for that matter. So yeah, they have this particular, the gut of the earthworm has ideal temperature, pH, oxygen concentration, all of that, and quite a number of good bacteria are also present. Um, I was supposed to. Right. What do we have first? Metamerism, right? Yes. I'll share my screen again with you. Thank you so much, and I apologize for taking more of your time, in fact. But then if we finish your portion, you'll at least know that this has been done, and it'll be easier for you to start studying for your examinations. So this is the next topic. Before that, I will just tell a little bit about the vermicompost. So basically, earthworm breaks down the biomass. So they are known as they act as bioreactors. This and then what happens is basically they produce enzymes that break down the biomolecules that are present in all the waste or the garbage into simple compounds that are used by the microorganisms. Oxygen that is produced by the microorganisms helps in various bioprocesses also, so that decomposition of the organic matter takes place faster. And the casting of the earthworm is useful in production of antibiotics, vitamins, plant growth hormones, all of it, in fact. So it's all in all, it's a very good thing if at all you have earthworms inside your the soil, so that irrigation at the same time, utilization and fertilization, all of it is taking place at the same time. Again, they have given you a little bit of vermicomposting. It is important to select the proper compost bed, and then you have a basal layer of broken bricks or pebbles. So it is there in your textbook. Please follow that 7.29 figure. You have around coarse sand, then layered by loamy soil. Then you have earthworms that is present in this above the loamy soil. Then you have the cattle dung or cow dung, and then you have the hay. Then you have the broadleaves and organic waste that is present, and then you have a net on the top so as to prevent the flies to come over. So this, if it is present in this manner, slowly what will happen is you start adding water to it, and then after stopping the water, this whole thing will be what do you call it, harvested, and not much damage is done to the worms basically. So the compost that is formed is finally soft, spongy, sweet smelling, dark brown in color, acts as a very good fertilizer for the plants. And where is it exactly used? So it is an excellent organic manure, porous, moisture. It has a wonderful moisture absorbing capacity, increases the uptake of nutrients, and then you have a huge market. In fact, if you Google it also, you get to see that you have earthworms and earthworm castings that are available commercially. And live earthworms are again used as fish bait, for especially while fishing. So they can be dried or processed also. And then they are also used as soil additives or animal or fish food. And this is all there. Now, when it comes to, we are done with earthworms right now. So when it comes to metamerism or segmentation, you get to see that it is the division or the segmentation of the body parts. As you can see, this is how metamerism looks like: segments or somites or metameres, and the animal is said to be metamerically segmented. We have seen all this, right? So differentiation of the body along the longitudinal axis into a series of similar segments or repeated segments or metameres or somites. This was first seen in animal kingdom, and they are externally divided by annuli, internally by septa, and there is a repetition of all the homologous parts, that is muscles, setae, nerves, blood vessels, etc. And these segmented structures are all interdependent, and metamerism is seen in certain arthropods and vertebrates also. So external metamerism is seen in certain arthropods. Internal, as again, it is seen in our vertebral column, simple, and the musculature. So in annelids, the metamerism is both internal and external, marked by constrictions on the body wall and annuli. Internally, it is marked by the transverse partitions. So there is a complete metamerism that is seen, and incomplete metamerism. These are the two types that are present basically. And when it comes to that, you get to see that incomplete as seen in the annelida, both internal and external, metamerism, it is the same basically. There is a repetition of the same structures. Whereas in incomplete metamerism, what happens is the body parts and segments are not similar. So it is seen in the arthropods, wherein external metamerism is seen, and in chordates you have internal metamerism. So that is how it is. And when it comes to the theories on the origin of metamerism, you have a pseudo-metamerism theory and you have a corm or a fission theory. And as such, in the pseudo-metamerism theory as proposed by Hyman and Goodrich, this has the metameric organisms that have been developed from acoelomates and unsegmented ancestors, having a serial repetition of the body parts. And this evolved as a result of accidental appearance of the transverse partition between the repeated body parts, so that each segment receives a repeated part of each segment. Metameric segmentation was linked with the evolution of coelom. So when it comes to the cyclocom or the fission theory, again, you get to see that. Please give me a minute. This segmented animal is a chain of coordinated sub-individuals formed by repeated transverse fissions or by asexual budding or some sexual non-segmented ancestors. And these sub-individual zooids, they remained united and became physiologically and structurally integrated so as to form the complete organism as such. Then you have the cyclomerism, basically. I just come to that. And then you have an embryological theory as well as a locomotory theory. So again, in the one that is given in your textbook, that is cyclomerism as proposed by Sedgwick, enterocoelom, this theory assumes that metamerism developed from an enterocele. So the enterocoelom had 4 gastric pouches. Do I have that picture? Unfortunately, no. No. Anyway, please listen to me. You have that diagram in your textbook, figure 7.31. You can just have a look at it. So, actinozoan coelenterate with 4 gastric pouches or enterocelic pouches developed from the gut or the digestive cavity. Out of these, division of 2 pouches resulted in 3 pairs of coelomic cavities called as the protocele, mesocele, and the metacele. And such an ancestral coelomate is referred to as a protocoelomate animal. And then unsegmented coelomates like mollusks and spinaculets resulted because this protocele got lost. So as such, this is how the theory was given out, but you do not have a concrete evidence regarding the same thing. And as I told you regarding the COM or the fission theory, they have told that, it is an individual formed by repeated transverse fission, and they remained united from end to end and became physiologically and structurally integrated to form the complex organism as seen today. And then the embryological theory, like I told you, is that, mesoderm during elongation of the embryo or the larva resulted in the mesoderm fragmentation, leading to repetition and of all the mesodermal derivatives in the adult. And when it comes to the locomotory theory, again, adaptation to locomotion, because of which you have the swimming, creeping, burrowing, etc. coming in. And it is a combination of the pseudo-metamerism, embryological theories. And in response to the swimming, and that is, undulatory, serpentine, that is creeping, burrowing movements, this theory had been postulated. Again, there is no particular or concrete evidence. So when it comes to the metamerism as such, you get to see that in different groups, you have different kinds of metameric opinion as such. So you get to see that it is in response to the body size, the locomotion, coelom, and evolution as a hydraulic skeleton. So according to this R.B. Clark, he had given that annelids arose as an adaptation for burrowing. So development of the septa, metameric segmentation resulted in the evolution of compartmentalized coelom, localizing the function of the hydraulic skeleton. So this helps in the bending movements of the body. So high structural development gives the scope for more evolution as such. And this is all. And then if you see, according to the significance, You see different scientists had postulated that proglottids are not addition to the body parts, but they are parts of the body as such with a specific function. So a series, so that they carry a series of organs. And then metamerism in different groups need not be similar to one another, but they have evolved independently from different groups during the course of evolution, so as to satisfy the needs of the animal according to that particular function what it was necessary. And as such, the significance of metamerism, as you have already seen, this helps in the locomotion, muscular action, fluid coelomic compartment, swimming, creeping, and then they have a high, this also shows a division of labor between the segments, because of which you have each metamer for a particular function. Example is the clitellum. Clitellum of the earthworm is specialized only for cocoon formation. So you also have pseudo-metamerism and nephridia that is given. There is one more slide. Please don't leave. Let me finish this. We'll finish this particular lesson. Nephridia and Celomo Ducts. Right. Right. So again, pseudo-metamerism is wherein, it is seen in cestodes. Your tapeworms, it has already been seen. So differs from the true metamerism in the following, that, the number of proglottids are not exactly fixed. So the number of segments are not fixed. Throughout the life of the cestode, that is the tapeworm, basically, you see that there is addition of new proglottids. Region of proliferation in the tapeworm is anterior, that is right behind the scolex. And then from then the age degree differ. You have the mature, immature, mature, and the gravid proglottids. And then the proglottids are again self-contained units having both the sex organs and excretory and nervous systems within themselves. So they are productive units that can be detached also. So that is why a tapeworm can be considered as a linear colony of incomplete individuals. So when it comes to nephridia, again, the nephridia are ectodermal in origin. They're segmentally arranged, coiled tubes, and they're internally ciliated, that is with the presence of a cilia. And they project as pushings into the coelom. They're closed or open. Closed means blind they can be, or open means they open into the coelom through a funnel. The ciliated funnel is known as the nephrostome, and the nephrostome may open into the coelom of the same segment in which the nephridium lies, or into the preceding, that is the before segment. The before segment. So this unit O is over here, but it opens into the before segment. So fundamentally, the nephridia are excretory in function, also have reproductive products to the outside. So they have reproductive function also, and they play a part in maintaining the salt water balance, that is the osmoregulation. And there are different kinds of nephridia present. They are classified basically on the fact on the basis of whether they are opening internally or externally, that is protonephridia or metanephridia. So whether they're open internally or closed, that is protonephridia or metanephridia. Whether they open to exterior or into an enteron, so that is known as exonephric nephridia or enteronephric nephridia. The name itself suggests. So please pay emphasis on the names. The names give you where they are located. Depending on the size, again, you have the meganephridia or the holonephridia, and the micronephridia or the meronephridia. And depending on the position, you have integumentary nephridia, septal nephridia, pharyngeal nephridia, and pre-testicular nephridia and testicular nephridia. And they also have a digestive function with peptid nephridia present. So different kinds of nephridia, as I told you. So you have a protonephridium also present. So this protonephridia is considered to be a primitive one and internally closed, made up of syncytial cells, that is without cell walls. And it ends as blind tubules at the end they end in the coelom. And the closed end of the nephridium has solenocytes that may occur singly or in groups. Solenocyte is a round ciliated cell connected to the nephridium through a thin duct. Is it present? Not exactly. You can see that in your textbook basically. Good picture is given. And excretory substances enter the tubules and through the walls by the action of this flagellum are sent out through the nephriospores. Thus, the protonephridium is open only at one end. So, example is that adults like adult polychaetes, like Vanadis, Phyllodos, Glycera, Tomopteris, Nephthys, etc., have this kind of protonephridium. When it comes to metanephridium, again, it's an open type of nephridia that is present, and the inner end of the nephridium has a ciliated funnel. Nephrostome opens into the coelom. This penetrates through the septum, enters into the succeeding segment. It opens through a nephriospore, and a metanephridium is open at both ends. This is the nephriospore, open at both ends. Example, Neanthus, Lumbricus, Polygodius, Protodrillus. And you have exonephric nephridia, which is also called as ectonephric nephridia, open directly through the nephriospore to the outside. Examples, Neanthus, Lumbricus, Ferretima, Hirudinaria. All of these have such kind of nephridia. You have enteronephric nephridia, open into the excretory canals or the alimentary canals, but they do not open directly outside. So this is used for water conservation. Example is the septal and the pharyngeal nephridia of the earthworm, Ferretima. You also have meganephridia or the holonephridia, which are large. Around one pair is present in each segment, and these open outside through the nephriospores. Again, this is seen in Nereis and Hirudo. Micronephridia, small in size, numerous in segment. They do not have a nephrostome. This is the nephrostome, as you can see, and but they open outside through nephriospores. So though internally closed, they are not protonephridia as solenocytes are not present. Example is again Ferretima. You have integumentary, means skin, no? So they are attached to the inner surface of the body wall. Example is again Ferretima earthworm. Septal nephridia, they are attached to the transverse septa as seen in the earthworm. Pharyngeal nephridia, protesticular nephridia, testicular nephridia present in the respective segments. Again, mostly it is either in the earthworm or in the leeches. We have seen pre-testicular and testicular nephridia in the leeches, that is Hirudinaria. And you also have peptonephridia, whose function, they function as digestive glands, and they are present in Ferretima, basically, the pharyngeal nephridia. So one or more types of nephridia can be present in the same animal in the same segment. And there are, so example is again Hirudinaria. All the nephridia in the leeches are metanephridia, but pre-testicular nephridia and the testicular nephridia are from segment 6 to 11 and 12 to 22 respectively. And in earthworm, that is Ferretima, all are micronephridia, and integumentary nephridia that are present are from the 7th to the last. So you have closed type exonephric nephridia. Again, the definitions of it, you will have to see what exactly are exonephric, enteronephric, closed, open types. Please pay emphasis on that. And then you have the septal nephridia and the pharyngeal nephridia also in the earthworm. We have just read about it. And then you have serpula, which is a tuberculous, that is, it's in a tube shaped. Polychaete. Nephridia show division of labor, that is at the anterior region, they are excretory in function. Posterior region, they act as gonoducts. Now let us come to something which is known as Celomo Ducts. They are wide tubes which are from mesodermal in origin. Open one hand to the exterior by the genital pore and on the other into the coelom by a ciliated funnel, which is known as the Celomo stone. So they so this function as secondarily as excretory organs, primarily as a genital what do you call it for genital ducts basically. And in earthworms, the reproductive funnels, that is ducts of the both male and female, are known as the Celomo ducts. And the urinary ferrous tubules of the vertebrate kidney are also known as the Celomo ducts. So Celomo ducts can be separate or they can be united with the cilia. You also have nephridium. So this is how it is basically. This is a nephridium, as you can see. Nephrostome, this is the nephridia Nephridiophore. This is the These are the types of nephridia. Protonephridia, which is closed. Metanephridia with a nephrostome. Nephridiophore. This can give you a better explanation of the diagram. Just look at these diagrams, you'll get the you'll be able to understand difference between micro and mega nephridia, and exo and enteronephridia. And we are here nephromixia, wherein you have a union of nephridia and Celomo ducts both together. They may be fused completely or partially, and that is with the nephridia to form the nephromixia. So they have both ectoderm and mesoderm. So you have both functions, excretion and excision of gametes. So again, in this, based on the kind of combination, you have a protonephromixium, that is a Celomo duct, plus protonephridium, that is both together. So it has both excretory and reproductive products releasing to the outside. Example is Phyllodoce. You have the metanephromixium, Celomo duct plus metanephridium, as seen in the Hesione. And mixonephridium, Celomo duct plus nephridium, and funnel is by the Celomo duct is by the nephridium. This occurs in Erenicola. Good pictures, good diagram has been given in your textbook. You can easily follow that. And then you have ciliated organs. Celomo ducts are reduced to these ciliated organs, as seen in Nereis. So they are attached to the dorsolateral longitudinal muscles and they open externally. So again, excretory system in Ferritima, it's been given. You have a good picture once you see the earthworm. This is all about today's class in summary. So I hope you have understood Annelida as such. And any doubts, you please let me know. And thank you for, I'm so sorry that I had to take your extra time, but unfortunately the next 2 classes also are going to be long. And I will have to ask if it is okay with you. If it is okay with you, I will take an extra class, because I've got to complete Arthropoda. You have Palemon. Hopefully I'll try to complete it in 1 class. And the whole of Mollusca and Echinodermata is there. So if possible, I will try to finish that also in 1 class. But then again, each class is going to be for 2 hours. If it is okay with you.
Okay, madam.
All right. Otherwise, 1 more extra day will have to be taken for me to finish the class.
Okay, madam.
So is it okay if I take an extra hour altogether?
Yes, madam.
I want everyone to answer, please. If you're not comfortable, please text me. I think I'm there in your group. But otherwise, if you're okay with it, I will try to finish the Arthropoda and Palemon in 1 class, and Mollusca and Echinodermata in 1 class. That is again, we'll have to see. And I might have.
Madam.
Is it okay?
Yes, madam.
Okay,
Is
then.
it
Thank
enough?
you. Okay.
Yeah.
And I will share the PPTs.
Yes, ma'am. Right now.
You want it right now?
Yes.
Okay, done. I'll try to share them with you. Okay.
Yeah.
Right,
Okay, ma'am.
yeah. Thank you so much again. And
Thank you,
happy
ma'am.
weekend. Thank you.
Yeah. Thank you.
Thank you.
Class